Going over the labs for this week, that I've only gotten one email about having any trouble, so I don't know if people are having trouble or just aren't remembering what I asked you to do and getting in there yet. But it's the bioethics lab, so you should be able to get to it from your lab menu. It should be active now. Let me open it up real quick. And again, I know it's different sometimes for me to log in versus you to log in. There's all you guys. So let me get to the book and look at it that way. All right, so in the labs, bioethics lab, I have a link to an NIH website. It's right here. Doesn't look like it's live, but you can either type it in directly or cut and paste this link to the bioethics page of the National Institute of Health. Let me turn down the mic a little bit. All right. Is that better? Now, well, I don't hear myself anymore. But anyway, the bioethics page on the National Institute of Health website. This will just familiarize you with the issues of bioethics. There are quite a few there. It's not exhaustive necessarily, but it's just for you to see what I mean by bioethics. We're talking about being able to make decisions concerning human health and other issues of technology that affect human health or may affect us in the future, our longevity, not only our health, but our longevity, and things that we're going to be able to do that may challenge us morally and ethically and so on. So some of these issues have to be very carefully considered, and I want you to pick one, find a link to something about it that is new, something new. Again, if you can surprise me, I'll probably give you extra credit about it, but that happens occasionally. Send me the link that you find. I'm going to choose one, and I'm going to put it in a discussion board topic for us to discuss, and that's going to be this week's lab. Yes?
I saved one. I don't know if it's new or old.
Well, I haven't checked my email in the book yet, because I just got back pretty late the other night, but if I haven't seen anything in my own inbox, so if you've sent it to my Hoagb@UHD.
In the response.
Okay. Well, I haven't, like I said, I haven't gotten in here yet to look at what's in here, and I'll do that. But I just wanted to go over this to make sure everybody was aware of that. This is this week's lab. We didn't meet Tuesday. We won't meet in class today, but I do want everybody to send me a link or put a link in here. If you send it to me in my Hoagb@UHD email, I'll get it. I'll check Blackboard in case somebody sent me that way, and I'll check my my topics in here, and I'll check this in here and make sure. But I haven't had a chance, again, after returning. I had a family emergency type of thing that took me away for a couple days, and again, got back pretty late, so I'm still trying to recover a little bit. But I will get in there, and what I'm hoping for is that I have all of the links from you by the end of this week, by the end of, really by tonight, hopefully at midnight or so that I can pick one and put it in a discussion board, and then I will let everybody know that the topic is now on the discussion board, and I want everyone to comment on it. Now, it'll be similar to the discussion topics that we already have. The comments I'm looking for do not have to be long, but they need to be well considered. All right, I want you to contribute to the conversation. I want you to have followed the link that's provided and understand what's going on there. And if you're the first one to comment, then you get to sort of take whatever side you want. If you come in afterwards, I want you to read previous comments and add to the conversation. So it shouldn't take too long. I mean, most of the time this should take will be finding something new and unique to send me. The comment that you leave in the discussion board shouldn't take that long, but it is part of the lab as well, okay? All right, so we are going to, I'm going to assume everyone understands this, and I'll be looking for these links again either later today. Now, I have almost all the tests graded. Again, I couldn't take them with me, so I have about maybe 10 or 15 left to grade. I'm going to do that this morning. If you want to come by my office this afternoon and get your test, that's fine. I understand that it's a Thursday and you won't see me again until Tuesday. If you're nervous about it, you can swing by and pick it up. If not, I will have them ready, no doubt, by next Tuesday. Really, like I said, I've graded all of the multiple choice portions. I just need to get to about 15 more of the take-home ones and get those put together, and I'll be ready to pass them back. Okay? Okay. I'm going to use two of the PowerPoints this time that I have used in previous classes. We're going to sort of try to cover both the genetics and the mismeasure of man. Today's topic is mismeasure of man, but since I went here Tuesday, the genetics topic from that. And they do go together. So what we're talking about, starting with this, is we're really starting to talk about our own biology, our own issues with human biology and health, and how this all works, again, in a societal sense, what the historical context of all of this is. And we've sort of started this when we talked about evolution, and talked about the modern synthesis. What was significant was the discovery of the structure of DNA, how it works to code information about our biology, and pass that information on from generation to generation. That's genetic, that's inheritance. The laws of inheritance that were discovered initially by Gregor Mendel, by growing pea plants, were rediscovered later on, and are now part of what is accelerating the change in biology that is really revolutionary. We are experiencing right now a revolution in biology that is similar to any other revolution we've experienced scientifically. The communications revolution that ushered in the internet and all of those changes, the nuclear age, which was ushered in by the discovery of nuclear, or the ability to accomplish nuclear fission and fusion eventually. The Human Genome Project, which was finished a few years ago, is starting to pay dividends in terms of new treatments, new drugs, quite a few of which are in trials, clinical trials currently, and may hit the marketplace sooner or later. This takes a long time to get something approved for actual use in the marketplace. It takes generally about 10 years to go through all of the trials that are necessary to make sure that it's safe for people to use. Now there's additional issue. Even with 10 years of studying, there are a lot of things that sometimes come up later that we didn't expect. And that's part of what bioethics is all about. Right? If you're a company and you're developing a new drug for the marketplace, and you are required by law to go through studies, there are occasionally mistakes made in those studies, or there are biases that influence a study. They're supposed to be double-blind studies, meaning nobody knows who's getting the treatment and who's not, not even the doctor who's administering the treatments. Only the statistician later on knows who got it and who didn't, so that they can determine whether it was effective, whether there were side effects, and all of those things. But there are still things that we don't expect sometimes. And again, we'll talk a lot more, if we could meet in lab, we'd spend a lot more time talking about this, but I want to develop some of those things in the discussion, and I'll participate in the discussion more than I have been with the other discussion topics, because this is a lab topic that I want to get across a lot of the points about these ethical issues. But we'll also have some time to talk more about it in next week's lab and going forward. But I want to try to get through, again, some of the basics of genetics. So these are the things, the laws of inheritance that we have, as scientists, we've sort of figured out by experimentation. Again, started with Gregor Mendel. Gregor Mendel was a monk who grew peas, so I'll use peas as an example. What was unique about pea plants is he really was able to isolate some very specific genes that occurred on different chromosomes. There's about 4 chromosomes in a pea plant, and there are some of the traits that are linked to those specific genes. For instance, color of the pod, color of the seed, whether the seed is smooth or wrinkled, things like that. And so simply by crossing plant after plant after plant, getting thousands of crosses, collecting all the data and looking at the patterns, he found ratios that you don't necessarily see with only one plant crossing. I mean, if you think about your parents, or if you have kids, you know, your nephews, nieces, whatever, you can see a lot of traits, but there's 2 things that I want you to understand. Nothing in our own genetics is this simple, for one thing. There's not a single trait that you have, not even hair color or eye color, that is this simple. Eye color is as close as we can get, more or less. If you have a brown-eyed parent and a blue-eyed parent, depending on whether the brown-eyed parent is homozygous or heterozygous, and we'll get to those terms in a second, you either have a 0% chance of having blue eyes or a 50% chance. So we'll go over that. But even there's, you know, my eyes are hazel, for instance. There's additional genes that can impact just how blue or just how brown, or whether they're in the colors in between, so forth. So it's nothing in our own biology is this simple, which is what made Gregor Mendel's work really sort of unique, in that he's somewhat stumbled upon an example of an organism that made it possible to see these ratios given enough crosses. Now, modern day, like you may have used fruit flies in middle school or high school to study genetics, fruit flies similarly have a few chromosomes that have specific genes isolated on some of those specific chromosomes. So they too can be studied in this kind of simple way. I'll emphasize this again about our own genes in a little bit, because a modern understanding of genetics is extremely complicated. I'm going to simplify it as much as I can. What we mean by F1 generation is that what's in the box are the odds of getting certain traits from the parent generation. In this case, we've got 2 green pea pods in the parent generation. But we know that they're both what are called heterozygous. You see the big G and the little g? We're using these letters to represent that gene that has 2 different alleles. Alleles are different versions of a gene. So in this case, the big G says be green, the little g says be yellow. It's called the recessive gene. In this case, the big G, the green gene or allele, is dominant over that one. So if you get the big G and little g together, this is called the heterozygous because they're different alleles. You still have a green pea pod because the dominant allele determines the color. If you cross 2 heterozygous parents, these are the odds of what you might get as the offspring. If you get both big G's from your parents, you're obviously going to be green. You've got both dominant G's. If you get the big G from one and the little g from another, you're still going to have a green pea pod because the big G is dominant. But there's a 25% chance that you'll get the little g from both parents and be yellow. So 2 green parents produce a yellow offspring about 25% of the time. Now this is like flipping a coin. Just like flipping a coin, it's odds. If you have 4 children, let's turn this into eye color. Let's say we have 2 brown-eyed parents that are both heterozygous. So there's a brown eye dominant over a blue eye recessive. 25% of the time, that's going to produce a blue-eyed child. But that's the odds. The 2 brown-eyed parents could have 3 blue-eyed children. Okay? Or they could have no blue-eyed children. They could have 7 kids and never have a blue-eyed child. It's the odds are that they'll have 1 of 4 who will have blue eyes. So just like flipping a coin, you can sometimes get a bunch of heads in a row, but you know the odds are 50-50. If you keep flipping long enough, the ratio gets closer to 50-50. Same thing with these. If we grow enough pea plants over and over and over again, these ratios begin to emerge. But within a very small sample size, it's not automatic. Does that make sense? Now the second one, the more complicated one, is what's called a dihybrid cross. This is a monohybrid cross, that's a dihybrid cross. And it's just meant to show these two, what are called laws of genetics, which are violated quite often in our own situation. But by understanding these general patterns, then we can study how the violations tell us anymore. But by looking at two traits at one time, we see different ratios come up. And so we can look at how each gene is independently assorted from the other. So these odds, these ratios, in other words, the color of the pea is not connected to the color of the pod. They're different genes, and in this case they're on different chromosomes. We have quite a few chromosomes, and so obviously some genes are linked together because they come on the same chromosome, even though each gene is independently inherited, because which allele we get are somewhat linked, and we'll see that. But the law of independent assortment and the law of segregation tell us that each gene is uniquely selected for and recombined in subsequent generations. So I'm not going to go through all of this. Actually, I will want to, just as a pattern, I'll tell you what the ratio is if we look at the two things together. The one thing I do want to point out, each one of these letters represents an allele in what's called a gamete. So during sexual reproduction, the woman's egg and the male sperm have half the number of chromosomes that you have in your normal cells, so that you only get one allele from each parent. Otherwise, you would get way too much genetic information in the zygote, therefore the developing embryo, and it wouldn't work. So we're going to show you in a little bit how we take the original genetic information and you copy it and then split it up into 4 different gametes. The 2 gametes come together and then put the 2 alleles back together. So if we're looking at 2 traits, since each one is independent, we have to look at 4 possibilities for each gamete. So let's say a sperm cell and an egg come together. The sperm could bring either the big Y or the little y, and either the big G or the little g. So you could have these 4 possible combinations in that gamete. This gives you a 9 to 3 to 3 to 1 ratio. In other words, there are 9 green pod yellow pea plants. Yellow in this case is dominant over green for the pea color. Green is dominant over yellow for the pod color. You're going to get 3 green pod green pea. You're going to get 3 yellow pod yellow pea and 1 yellow pod green pea. Okay, so it's 9 to 3 to 3 to 1 ratio if both traits are dominant. It gets really messy when we have what's called incomplete dominance. I'm going to skip to that. Okay? So in this case, a heterozygous condition might give you a different trait, a third possibility. You could have parents that are red and white, but they're both homozygous for red and white. In other words, the white one has 2 alleles for white, the red one has 2 alleles for red. They can only give those alleles to the offspring, so the next generation is automatically going to be heterozygous. They're all going to be pink, right? Because they're all going to get 1 red, 1 white allele. That incomplete dominance produces an intermediate trait. Now, if you took 2 pink flowers and crossed them, then you'd have 1 red, 25% of the time, 1 white, 25% of the time, and 2 yellow, pink. Those are the odds, anyway. Okay, but if you took, in this case, red and white flowers, you would get all pink in the next generation. That's all you could get. If you cross 2 pink, then you have the odds again of a 1 to 2 to 1 ratio instead of 3 to 1 that we saw in the first one where there's dominance. And it really messes up the dihybrid cross. I can't remember, we'd have to look through it again to see all the different outcomes, but it's 1 to 2 to 2 to 3 to 2 to 1. Anyway, it's just not a mess, it's a predictable ratio, but it's much more complicated. All right, let me go back and talk a little bit more about what I talked about, or referred to what's called meiosis. And again, these should be somewhat familiar terms, right? You studied this in high school. And I'm not trying to go back and again expect you to remember it all, but just to keep it in our understanding of the way we're talking, because the points I really want to make have to do with what we're about to get to is what is a gene, right? I want to talk about what is a gene. because I have a feeling that most of us from high school get a pretty simple picture of what a gene is. That may not be real accurate. But looking at meiosis again, you take two normal cells, somatic cells. In this case, they're called germ cells because they're going to produce gametes, but they have a complete set of chromosomes. The first thing that happens during meiosis is you copy them. You copy every chromosome, so now you have twice the number of chromosomes that you normally would have. Then you pull those homologous chromosomes apart, okay, the copies, which are homologous because they're the same. You pull them apart into separate cells. Then you take that chromosome, each of those chromosomes, and you pull the two pieces of it apart. Each chromosome is actually two chromatids connected together. You separate those out. Now what's unique is that each one of those those chromatids in the paired chromosome have one allele for each trait. They have alleles for the same trait on each one of those chromatids, and when they get separated, that means each daughter cell now at the very bottom has one allele for each gene. That's called the haploid condition because it's half the number of chromosomes as a normal cell would have. A normal cell has a diploid number. So this has to go through two different divisions to get to this point. When you grow, your cells undergo mitosis, which is similar to meiosis, only that it only does the first step. It doesn't go a second time. So you make a copy of your DNA, split it up into two new cells, and that's how you grow, but each new cell is diploid. In this case, we end up with haploid gametes, so that when we put them back together, we get a diploid zygote, or offspring. All right, if you look at a chromosome, it's basically a strand of DNA, really long strand of DNA that if it's all unwound, really is extremely long. Remember how long, but feet long if you undid all of your DNA. So it's really compacted up. It scrunches up so that when we look at a chromosome, we generally see it all scrunched up, and you can actually see it under a microscope. Then they look like little X's and occasionally a Y. The male chromosome, the male has a chromosome called a Y chromosome because it's missing a leg on that X and literally is missing those alleles that might give information for a trait. And we'll talk a little bit about those sex-linked traits in a minute. Where a certain sequence of DNA is an instruction for your cells to do something. And so when your cell reads that sequence of DNA that says make this enzyme, or turn this gene on, go read this other part, or turn this on, or turn this off, or all kinds of different types of instructions. That's what your cell does. We can identify where those sequences are that give us specific instructions, and we call those genes. There's a particular location on the chromosome where those genes are found, which is called the gene locus. And there's a certain percentage of times those genes appear, the gene frequency. And we can think about that in a population sense as well. The gene pool is all of the genes. For instance, if we were all the people on the planet left in this room, let's say something happens while we're in class and everybody else disappears, we're it, right? We're the gene pool. This is all the genes that we're carrying is our gene pool. Now, that's not the case, so we could talk about the gene pool of Houston. Let's say nobody could ever leave or come into Houston. Houston was a domed city. Well, then our gene pool would be all the genes in Houston. Now, we have a global gene pool now. People move around all over the place, right? People migrate and immigrate into and out of populations. So we have a global gene pool that's constantly being somewhat mixed. We occasionally see certain alleles concentrated in some parts, some areas, because of the gene frequency is varied. But overall, we can mix it all together. So the concept of a gene pool is something worth thinking about as well. All right, so let's get to what I mean by a gene. Again, I want to just be specific about what a gene is. And this is some of the rest of the just vocabulary that comes along with this, and I know it can get confusing. But when we look at what the alleles actually tell us, that's the genotype. So the big G, little g, genotype. But that doesn't tell us exactly what trait is actually produced. It could be, it depends on whether there's a dominance or an incomplete dominance, right? So the phenotype, what we actually see, is not exactly the same as the genotype. We have to understand both to really understand how the genes are working. All right, so as we go forward, we'll talk more about, I have genotype and phenotype up there twice, but I didn't. All right, so we've seen this, and this is my demonstration of that. We can have the heterozygous genotype. If there's dominance, it would be red. If it's incomplete dominance, it would be pink. It's the same genotype, but we can get different phenotypes from it depending on dominance or not. So, we used to sort of teach one gene, one enzyme. In other words, an enzyme is a molecule in your body that controls a chemical process, a chemical reaction. For instance, how to make proteins, how your body makes proteins. And it seemed for a while as we were studying genetics, that was a good model, that it made sense. Because we know that a gene doesn't specifically instruct for a phenotype. Phenotypes are very complicated. They take more than one gene to accomplish. Like I said, even eye color, as simple as it is, has multiple genes that can influence the color of your eyes. And anything beyond that generally takes many genes, maybe hundreds of genes, working together, providing different pieces to the puzzle to actually produce a trait. And traits can be anything from how tall we are to our personalities, okay? How quickly we get angry, or or even some of our, how much we enjoy being afraid. Right? We've got Halloween coming up. Some people like getting scared. Some people don't. There's a genetic basis for those things. Okay, now they're developed within an environment. So you can learn to be more afraid or learn to be less afraid, those kinds if they're flexible. But that's the benefit of having a trait that is the result of many genes. It makes that trait more plastic, more flexible. If you have a trait that's just so fixed, so hardwired that you can't vary it to the environment, then it's probably not a good gene. It's probably not going to allow you to adapt and survive. But we realized fairly recently, which is decades ago, that it's not that simple. So then we thought, well, maybe one gene, one polypeptide, because a polypeptide is just a segment of a gene, right? It's just one of those amino acids. So the gene says this, still not that simple. We have found, when we look at all of the genes, that there are any number of different types of instructions that the genes can provide, such as, go to this part of the genome and get this information. One gene may say, go read this part, or cut this one out, or put this one on. So turning them on, turning them off, how long the gene is being copied, how many copies does it make of something, those kinds of things. All are controlled by our genes. And there's a couple other things too that we've found as we look at these genes, and one of them that I think is becoming even more important, that we're starting to understand even better, is what's called epigenetics. And this is the influence of our environments on our gene expression. It starts even during development, in the womb, during fetal or embryonic and fetal development. There are genes that can be turned on or not, depending on your environment. So it could be your mother's diet, or some other part of what's going on. Even after birth, this can happen. If you get nurtured, that turns some genes on. If you don't get enough nurturing, those genes don't get turned on. And this turns out to have quite a bit to do with our personalities. It's a somewhat tricky thing to talk too much about, right? Because I don't want to give the impression that anybody who is sort of antisocial. It's all their mother's fault because of her not nurturing them enough. It's not that simple, but there is evidence that we're seeing going on that those influences do have a lot to do with what genes get turned on and which ones don't, and that's called epigenetics. So again, if you're interested in this, you can look into this a little bit more. Even though that's true, again, let's say we could connect certain personality traits to a lack of nurturing. You still have control. Again, keep in mind, these things are still plastic. Not that the gene can be turned off or turned back on, but the trait itself is complicated enough that you can use training and other environmental tools to sort of correct for it or overcome that or compensate for that. So if you think about addictions, how susceptible somebody is to becoming addicted, that doesn't mean they're going to become addicted. They can learn to deal with it. It's not easy, but it's possible. So you're not doomed by your genes, but we have every variation across the board of all of these different traits within the population. Within the gene pool, for any trait, there is a wide variation of possibilities. I've been throwing around the term development a little bit. I got to speed up here pretty quick. So just real quickly, the development is, you put the two gametes together, you get a zygote. That's one cell, single-celled zygote. It starts splitting into 2, then 4, then 8, then 16, and so on. You eventually get a sort of ball of cells called a blastula. We can follow this development. It's interesting, if we follow our own development, there are points at which we have a tail, there are points at which we have gill slits. This is called ontogeny recapitulating phylogeny. If you want to look that up, it's a funny term. But it's just sort of the fact that the pattern of our development seems to follow the pattern of our evolutionary history. Now, that's not a scientific, we can't test that scientifically, but it's a pattern that, again, why we have gill slits at one point of our development, why we have tails and so forth, is an interesting question. You can't really distinguish our embryos at different stages from the embryos of any other animal or chick or anything else. Now, once you get past a certain point, then they all developed beyond that. But early development is the same for all organisms and diverges later on. One of the key stages is when the blastula gets big enough, it begins to invert on itself, and so you get these 3 layers of cells that are forming a gut, and then we have an internal and external tube within a tube system, and that forms during the gastrula stage. Beyond that, it becomes an embryo, eventually a fetus. Once you're born, you're not done developing by any means. You're not done developing until you're about 20, I've seen 24 to 27 or so. And then you start to die. Sorry about that. I'm in that stage of my development, senescence, going backwards. But, and if you've raised children, you see this. And really, the adolescent mind is still changing, it's still developing, and it does so until you're about 24 to 27 years old. We don't always think about that as we're growing up. We know we stop growing physically, but we're still developing a lot of these traits that I'm talking about through adolescence into adulthood. And even in adulthood, again, those traits are flexible. But influences in the environment during our adolescence can have a big deal to do with, for instance, an adolescent brain is not fully developed. So if an adolescent's doing drugs, they're messing up their development. They're not just getting high. They're telling the brain, oh, stop pruning cells, stop becoming smarter. Literally, things like that are happening even during adolescence that have a big impact, our environment having a big impact on our biological development. So I'll go into, again, there's a lot I could go into there, but and schools are starting to introduce a lot of this information now to teens so that they can make better decisions. Like if you go smoke this pot or drink this alcohol, it's affecting your brain, right? It's not just a temporary thing. All right. So I've sort of been beating around this idea. How important is genes and how important is environment? What's more important? We've had this debate forever. It's called nature versus nurture. How much of you is just genetically programmed. There's not much you can do about it. How much of you has to do with your parenting or the TV shows you watched growing up, or your peers' influences. All of these environmental influences, they all work together. And it's interesting when you look at this because we can find some traits that are very genetically determined. We can look at percentages of behaviors in populations, for instance, and see these bell curves of distribution. And whenever you see this bell curve of something, you can pretty much guarantee there's a genetic influence there. And most of us are going to be within the standard deviations of that mean, but there are going to be a few people who are just really far in either direction, and they're either really rebellious, or they're really meek, or they're really outgoing, or they're really sort of stay to themselves. And most of us are somewhere, again, within this sort of standard deviation of a mean. There is no one, there's no person who's normal in that they're right perfectly on the mean for every trait. It just doesn't happen. Every trait has this bell curve to it. And that's genetically determined. But again, what you do with that, right, because I'm an introvert, but here I am, right, sort of bouncing around up here. I can be outgoing if I want to be, right? So I'm not doomed to sitting in my room and reading books all the time. And so the other thing to point out, and again, God, I'm not going fast enough. Every trait, though, that we talk about is really more than one gene. I've said that enough times, I don't have to do that. There are some traits that have multiple alleles, just real quickly, like our blood type. You have 3 different alleles. You can have, blood type is what antigens you have in your blood that help your immune system identify foreign things. These antigens can either be A or B or neither, which is O, and you get alleles for each of these 3 things. So there's 3 alleles there. That's multiple alleles instead of just 2, as we said. There's also sex-influenced and sex-linked traits. These are a little bit different. Again, because the male chromosome, the Y chromosome, is actually missing a piece, there are some things that occur on the next chromosome that can't get matched by the male, so they're linked. And things like baldness, certain types of baldness, the royal family, the classic example of this is hemophilia in the royal family, passed on from generation to generation because of all the inbreeding they were doing, right? They didn't have a very big gene pool. It was just the royal family in France and England or wherever, and they kept sort of marrying cousins, and it led to a lot of this. Anyway, so I won't go into a lot of that detail about that either, but it's all interesting to think about, particularly if you remember doing this in your high school classes. Mutations can happen in a number of ways. You can have deletions, for instance. Now, if that happens to your cell when you're living and it's not a germ cell, it's not going to become a sperm or egg, that can lead to cancer. So this is why we take antioxidants. It's why you try to avoid eating charred foods, things that have free radicals into your system that can cause this kind of mutation and lead to a cell not functioning properly. If it happens to a germ cell, it can lead to birth defects or the embryo not being able to develop at all. Now, a single deletion generally doesn't cause as much damage as something else, like a duplication. If during the copying of your DNA, you copy genes twice, well, then your cells aren't going to function properly. You can have an inversion where they get sort of switched. Then the instructions aren't going to be in the right order and things can go wrong. A translocation or even a there are situations, I don't know why it's not on this list, where, for instance, in trisomy 21, which is Down's syndrome, there is 3 chromatids attached instead of 2, and so you get all that extra information for a lot of those traits, and it leads to developmental differences. These translocations and non-disjunctions, anyway, where you might get a piece of that doesn't get passed on. So not just the Y chromosome, but another chromosome is missing information, or it has twice the information anyway. All of these things can cause problems during development. There it is, non-disjunction. So non-disjunction, again, just as an example here, we end up with 3 copies instead of in 2 of these gametes, only 1 in the other. You should have one in each. So this whole chromosome isn't even occurring over here. Most of these kinds of things are really severe birth defects. And there are some of these that happen often enough to be named as a syndrome, like Down's syndrome. But there are also many of these that happen rarely, but most of us probably have a relative who's been affected by something like this in some way. All right, I'm going to skip over viruses because we talked a little bit about them with Ebola and all that going on. I want to go to my second PowerPoint because I want to at least pose this problem. Genetics have been, unfortunately, used throughout our history in a very prejudicial and biased way to claim superiority for one group of people over another, for instance, based on genes. And it's been very unfortunate. It begs a question of some sort, just how susceptible are we to these kinds of xenophobic instincts? Because it's not limited to only one group of people oppressing one other group of people. I mean, it can happen anywhere, and throughout history has happened in almost every culture. And so it seems to be a pattern of behavior that is somewhat ingrained, that again we have to control. And it's been studied, and it's not just about racial biases. It's not just about the book, The Bell Curve, which was a book written, I can't remember, 70s or 80s, one of many. But it claimed, based on IQ scores and genetic differences, that Caucasians had superior intelligence genetically to African Americans. It claimed Asians actually had the highest IQs, and so on. And it tried to make it look scientific by referring to gene frequencies and all this terminology that I've been using. Completely debunked. But I've had these arguments with people in my blog. So people are still out there believing this stuff. It goes beyond that kind of bell curve thing to even men over women. Oh, men are smarter than women because our heads are bigger, right? Wait a minute, I'll scratch my head on that one. If that's true, boy, whales, they should be a lot smarter than any of us, right? It's all about brain-to-body ratio, which we know pretty well now. And even within that, that's just a very general term. It has a lot to, even in a smaller brain, you can have a higher IQ because, anyway, I won't get into all that detail. What I mean is, what I'm trying to cover here by the mismeasure of man, and this is a book that Stephen Jay Gould actually wrote, and I encourage you all to read it, about the number of cases of this happening. Even when people, immigrants were coming to Ellis Island, there was a lot of this kind of categorizing people based on the shape of their skull, right, into people who were expected to contribute more or less to society. And it was very unfortunate, and some of it was just outright racism, and some of it was ignorance, but all of it is wrong. So we can look at all these issues, and it's time to bring it up when we talk about genetics. Look for these. You can look for these kinds of things when you're looking for your bioethical considerations, right? This is in play with that, because even though it's not a direct application of genetic engineering or anything like that, it is a misunderstanding of genetics and misapplication of that understanding. Now, the last thing I'll talk about here are some of these new ideas, and I've touched on some of these. Like how much of intelligence is genetic and how much of it's environment. How much can you really learn to be smart? And there's a lot of flexibility there. Again, everything has a lot of flexibility. You can go out and get your IQ tested or whatever, and you get a number. What does that number mean? It means very little, right? It's just one indicator of how you think, how you process information. Multiple types of intelligence, multiple skill sets, all of these different things should all be assigned different things. So those are the kinds of things we're talking about that we have to be careful about. Are criminals born, not made? Right? Has anyone seen Clockwork Orange? something like that. I mean, again, this has been the topic of conversation in our society for a long time. Because some people are tempted to think, well, if we could find a gene for criminality, we could identify criminals before they commit their crimes, and we can, minority report them or something, right? We can get them out and avoid all this, pain and suffering in our society. All we have to do is look at our genes and find the criminals before. Well, come on, right? I hope we never get to that point. But we are getting to the point where we can identify genes that might influence criminality, just like we'll find genes that might influence weight gain versus not gaining weight. We might find genes that, and again, there's not one gene that's going to do that, that's going to control our weight. Many number of genes, but undoubtedly in the next few decades we're going to find genes that influence all kinds of things in our personalities. How genetic is a psychopath? I mean, how avoidable is that? Somebody just goes off, right? The green light up there protecting us from psychopaths who lose their minds and start shooting everybody up. Is that genetic or is that environment? How much of it is each? And we may start to unravel some of that, but we have to be extremely careful as we do so. One other thing that's interesting, I've mentioned this before, treating our genetic disorders like my celiac disease, which has a genetic component, that's a personal thing, right? I mean, there's a percentage of the population that shares that gene with me, but most people don't have it. But each one of us probably has something like that, where we have an extra susceptibility to something in our diet or in the environment. We're more sensitive to the sun, or we're more sensitive, again, to various interactions with drugs that we take. Some can tolerate Tylenol, some can't, those kinds of things. Each of us are unique. So we are finding those genes, and we are beginning to develop the treatment that takes that into consideration. So rather than having a drug that can be prescribed for a disease and anybody can get that drug, you'll have a drug tailored specifically for you. And that's something that's called pharmacogenetics that's on the horizon itself. All right. We've talked a little bit about what makes us human, and that's in here as well, but I'm over time already, so let me get to the quiz, and we can talk about some of this more as we go forward. Because I have moved that part of that old PowerPoint into, so we've already talked about that. I'm relying on my old PowerPoints a little bit, just because, again, I've been a little bit overwhelmed by some family issues that I haven't kept up as well as I'd like. All right, let me get to, now we're getting to the point now where, whoops. We're going to see a lot of things in this chapter. 13, chapter 13 is, we're going to stay in chapter 13 for a while. You also need to realize that we can pick one of the two quizzes, either the genetics quiz or the mismeasure of man quiz. That's the first one I'm going to get to, so we'll do this one. Don't worry about the fact that we're not going to do both in class, because we didn't meet on Tuesday. All right, this first question comes from something that we covered a while back. It's sort of interesting that humans are the only animal that can learn through direct instruction, the way I'm teaching now. Excuse me? Oh, no. You said hello. What's that? This isn't true? You can learn. Other animals can learn by seeing and copying, right? By example. Can learn by example. And maybe there's a subtle difference here, but what I mean is you can just sit there and hear me say something and learn without having to actually do it yourself.
Oh, that's what it means.
Right.
That's true.
Right. So there's no other animal that can, a lot of animals can learn by example. Right. So if I demonstrated something and said, okay, now you do it, well, that's not unique to people. What's unique to people is you can just sit there and passively hear me say something and, oh, okay, I understand that. It may be a little bit subtle, but it's an interesting difference. So that's true. Yes.
Well, first, I just wanted, have you ever heard of the psychoprobe, like in Asimov's books? Do you think we'll ever get to that point where we can, kind of weed out certain undesirable characteristics or change people's brains? And then also, I talked to you about handing out a questionnaire. I'm doing a class project that I would appreciate student input for.
Are you ready to do that today?
Yes, I am actually.
All right, let me pick up on that, because I know, again, we're 5 minutes over, so if anyone has to get to another class, let me finish this real quickly. Let me finish this quiz real quickly, and then if you would, please stay and participate in this survey that she is doing. We'd both appreciate that. The question about whether we're going to get to what Asimov was writing about is sort of yes, and we hope not completely, right? I mean, we've already started along that path. We're already doing some of the things when we take serotonin reuptake inhibitors and things like that. That's not necessarily a bad thing, right? That may allow us to be more of ourselves than if we had, because I mean, mental illness is an illness, and we should treat it just like any other. But where we find those lines, where we don't want to go over the line, when is it too much to medicate somebody for ADD or depression or something? When is it, finding those lines is what this week's lab is all about, right? Because as a society, we have to decide. Who gets to decide who has to take medication for depression? The depressed person or society or whatever, right? What if we find a gene that sort of makes somebody susceptible to being criminal, but we can give them some medication? Well, how voluntary does that have to be? Does society have the right to impose that kind of treatment? Those are the kinds of things we're talking about, and those are going to be things that are going to challenge all of us in the next few years. Epigenetics is the study of changes in gene expression during development and early childhood due to environmental factors. That is true. Oh, I think they don't always have true on the top, do they? Maybe that's why I've messed up a couple times. Human species may have evolved an upright posture in order to become better hunters. That's true. Again, I don't remember if I've talked about that before, but there's a lot of evidence of this, and you'll see at TED.com, for instance, at some point about how much we evolved to move, our movement. Craniometry is an effective way to determine a person's intelligence, simply by measuring the size and shape of your head. No.
That was true?
That was false. And the out of Africa hypothesis explains why all humans are so genetically similar.
True.
That's true, because we have a very recent divergence of cultures in different parts of the world. And now we're all coming back together again, and even some of those differences even those slight differences, very slight differences, are being addressed. All right. So, sorry to run over. Yes? Show you the lab again? Yeah. 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 Show you the